Here we have Vincent checking out his flask with the leftover HeLa cells from the passage. It's extremely important to hold the flask vertically when working with it in the hood, and you don't want any DMM getting into that filter. Always make sure to also spray your gloves and anything you'll be using the hood, especially if it's not sterile already. Here we are seeing Vincent label the cell suspension in our conical tube because we have already transferred it over from the flask. So he just labeled it cell suspension. Eventually, after we get our cell count, we will actually move that into a different conical tube with our dilution. So here he's just adjusting a P200 pipette to 20 microliters, as that is the recommended amount to pipette into our cell counter slide. He's going to pipette it into the small divots on both sides of the slide, so there's two little divots, and they work via capillary action, which you will see here. Right now he's just pipetting the cell suspension. Now you can see here he's pipetting it into one of the divots on each slide. It's very important to use both hands as it helps with stability because it's a very tiny hole that he has to pipette it into. Another thing to note is that you can also, in some cases, use a trypan blue staining with the cell suspension, which you can see in your protocol on benchling. However, here we actually just loaded the cells into the BioRad cell counter slide. So in the BioRad machine, the TC10 actually accounts for that already. So now you can see him pipetting another 20 microliters into the other divot of the slide here, and this will go directly into the BioRad TC10. Now we see Vincent getting up and he's walking to the BioRad TC10 where he's going to insert the slide onto one side of the slide into the machine, and he's going to wait for it to initialize, and the machine will count the cells on its own. From there, we get the cell count, we take note of that and write it down for our averaging calculation that we'll do later. Here, he just rotated the slide and also entered it in via the other side and takes note of the cell count, which we'll also use for our averaging. And now he's just returning the slide back to the hood, which we will actually discard.
I'm going to walk you through how to do the diluted cell suspension to make our reverse transfection. So in the video, you saw in the previous clip that we counted how many cells were in the suspension of cells. So that was just cells that we've passaged. We put the new passage in the incubator, and now we just took all the remaining cells left over, put them in conical tube 1, and we counted them just as they are. We put them on slides A and B, which are just the two sides of that little slip that you saw, and we put both into the counter. So in the video, we saw that slide A had a concentration of cells reading 4.50 times 10 to the 5th cells per ml. So this is a concentration. Slide B had a count of 2.92 times 10 to the 5th cells per ml. So again, just the cells that we're starting with, these are the two concentrations for both sides. And that's just because they read differently. So we can find the concentration or the average by just adding them up and dividing by 2. Give me a second to do that. (3.71 times 10 to the 5th cells per milliliter is the concentration that we now can safely say we have in that flask. So now this number here is going to serve as our concentration 1. So the concentration that we're starting with. And this is really just an M1 V1, M2 V2 equation. In class, you'll often see it as C1 V1 equals C2 V2, just for concentration as opposed to molarity, but it's the same thing. So our starting concentration of cells, again, is this 3.71. the V1 we are going to solve for. So how much of that in terms of volume do we need to take out to make our new cell suspension? C2 is the final concentration that we're looking for, and V2 is the final volume that that's going to take up. So in this protocol, and we'll display this in our video, we need to make a final concentration. I'll just write down the parameters over here. We need to make a final concentration of 20 times 10 to the fourth, also 2 times 10 to the fifth cells per ml. That's the final concentration we need. And that volume, which will be V2, that final volume, is going to be 0.5 ml per well plus 1 extra ml. So in this video, we had 3 wells, so 3 times 0.5 is 1.5, plus an additional 1 ml is 2.5. So 2.5 ml cell suspension. So again, just to clarify, we're starting with this concentration of cells. We are going to solve for how much of that we need to take. We know that the final concentration has to be this, and the final volume has to be that. So really, this is just a plug and chug. So again, C1 is 3.71 times 10 to the fifth cells per ml. How much of that do we need to take? Don't know yet. That's our first volume. The final concentration, like we listed up here, is 20 times 10 to the fourth cells per ml. And the final volume, like we said, is 2.5 milliliters. So now these units can cancel, cells per ml cancel on both sides. So you know your final units are going to be in ml. So I'll just take a second to do this math out. And the initial volume is 1.35, give or take, milliliters of original cell suspension. Okay, so now this is the volume of this concentration of cells that we're moving from conical tube 1 to conical tube 2. And I'll draw a little diagram just to make it clear. So you've done your passage. And you should have a flask of cells, roughly about that color, with the concentration of 3.71 times 10 to the fifth. From that, we're going to take what we just calculated, 1.35 milliliters of that concentration to make a 20 times 10 to the fourth cells per ml concentration. Now, this can't just be the 1.35, because like we said up here, our final volume has to be 2.5. So what else do we use to fill that gap? Well, we use just DMEM in this case. So to do that math, you're just going to take, we know that, again, our final volume is 2.5 milliliters. So 2.5 ml, which is our final volume, minus what we know we're putting in from here, 1.35 ml, is going to equal roughly 1.15 ml DMEM. Just to top it off, and I'll draw that out here. So that is how you make your final cell suspension. And then this is what we're going to use ultimately to do a transfection. And those calculations will be in the procedure.
With our cell suspension dilution calculation finished, now we can actually make the dilution in a new conical tube. So Vincent is transferring the correct volume of cell suspension into the new conical tube. So that's our old cell suspension from the passage. He's going to pipette it up in a serological pipette and transfer the correct amount into the new conical tube, which will be our dilution. And this number obviously will change based on your cell count and the average cell count you get from the Biorad TC10. Another helpful tip is to always loosen the caps before you're on the step of pipetting the cell suspension and the DMEM into the new conical tube. And another key thing is to always change your pipette tips whenever you're working with a new cell suspension or a new media, such as a new DMEM, which we will transfer into the conical tube. When pulling up the liquid with the serological pipette, make sure that the pipette is actually in the liquid, so you're pipetting up the correct amount. And also always make sure you have the right size of pipette, since you don't want to get it sucked up into the serological pipette. And here we just see him taking that pipette once he's done using it and putting it into the bleach beaker, which should be in every single hood you are using. And then here he just labels the new dilution that we'll use for the transfection.
This clip is going to walk you through how to figure out what volume of plasmid you need to add to your Jet Optimus buffer in the microcentrifuge tubes. So you just saw, this is a microcentrifuge tube. You just saw that we added to each tube 50 microliters of Jet Optimus buffer. To that, we need to add a certain amount of DNA. That solution is what we're going to pipette into our wells, on top of which we can then add the cells. So just again to clarify, so far we have this, and you saw that in a previous clip. Again, we have the 50 microliters of buffer that we added to every tube. And now on top, we're going to add some DNA. It's a very small amount, but this is going to be plasmid DNA that we got from the freezer of X volume. We don't know how much right now, but that's what this video is going to show you how to calculate. So those plasmids came out of the freezer. They have a concentration of 500 nanograms per microliter. Okay, so that's the concentration that we're starting at. And this is going to serve as a sort of conversion factor, if you will. So in our procedure, it says that we need to add to this, right? So it says that we need 0.5 micrograms. So how much volume is 0.5 micrograms? So we can do that calculation simply. So 0.5 micrograms. Since this is in micrograms and this is in nanograms, I'm just going to quickly convert. There are 1,000 nanograms in 1 microgram. So that just gets us into the unit that we want. So we can then use this over here, like I said, as our conversion factor. The nanograms has to be on the bottom to cancel out this, again, concentration of plasmid. The units will cancel, and I'm left with just a volume. So we can quickly do that math out, and it comes out to 1.0 microliters of DNA or plasmid. So again, just to recap, you have your 50 microliters of buffer that you have in every single microcentrifuge tube, the negative control, the clover, and the M scarlet. And now you need to add respective DNA to all three. You're not adding any of this plasmid DNA to the negative control. That's why it's the negative control. But for the D clover, you're going to add a microliter of D clover into that tube. And for the M scarlet, you're adding a microliter of M scarlet into that tube. And that's just represented by the orange on top of this blue. You're adding it to the buffer. And that's pretty much that step.
We see Vincent grabbing the Pnex and the D clover DNA from the icebox right before making the transfection mix. Here we see Vincent pointing to the DNA D clover, the Pnex. Now we have the negative control tube labeled, the D clover tube, and the MTS tube. Always make sure to have this laid out before your experiment. Here is the PJet buffer. This is what it looks like. And now he's showing you the PJet Optimus reagent. Now Vincent's using a 200 microliter pipette, and he's pipetting 50 microliters of the jet buffer into each respective tube, so the negative control, the D-clover positive control, and the P-nex tube. Here, changing the tip is always a good practice to reduce contamination in science. However, since the tubes are actually sterile and are in a sterile box before we put them in the hood, we actually can use the same tip here, especially since there's nothing in the microcentrifuge tubes. Here we can see Vincent actually taking our D-clover DNA. He's picking it up and he's going to pipette 1 microliter, which we calculated beforehand, to put into our jet buffer. So we'll do this first, and then we will do this for the P-nex DNA as well in the MTS tube. We don't add any of it in the negative control because that is getting no DNA or no plasmids. Always make sure to change your tip as well before going into a new tube, especially with new DNA, as you don't want them to be mixed up and contaminate each other. Now with the P-nex, he's actually using 0.5 microliters instead of the 1 microliter that we use for the D-clover, because it actually is relatively toxic to the cells, especially with the higher concentrations of DNA. Once you have pipetted the DNA and the buffer into the tubes, you will go over and briefly vortex each tube for 1 second, like Vincent is doing here. Generally, using 2 hands is much safer to hold the tube so it doesn't fly out. And then also, once you vortex it, you want to spin it down briefly. And it's always important to make sure that the microcentrifuge is actually balanced, or the mini centrifuge is balanced. And the reason we do this is to make sure the DNA and the buffer is all at the bottom of the tube and it's not trapped in the lid or the sides of the tube. Now we see Vincent here actually adding the final 0.5 microliters of the Jet Optimus reagent to our transfection mix. And again, it's really important to ensure the reagent is actually in the tube and also in the pipette, so we know we're actually picking up the reagent, or if you're out of it. So first, he's taking it out and putting it into the negative control, and then he'll respectively add it to the D-clover tube and the P-nex tube as well. Also notice how he's changing tips every single time he is pipetting the reagent into a different tube, because each tube has different DNAs and or different amounts of DNA, and we don't want to get those mixed up, as it'll alter our transfection results. After Vincent has put on the cap on the Optimus reagent, he is now going to do the same vortexing step for each tube, and also will briefly spin it down again, as we don't want any of the reagents trapped on the lid or on the side, and we want to make sure everything is at the bottom of the tubes prior to adding the transfection mix to each well. Here Vincent is showing us the sterile plate. It came in a plastic box and we took it out. And now he's labeling the wells according to each of our respective transfection tubes. This is important to do during the 10-minute incubation, as you have some downtime here and you can get yourself more organized so the rest of the protocol runs smoothly. So here he's just showing the initials in the date. There's our negative control in the second well, the D-clover in the third well, and the MTS P-nex. Since this is a reverse transfection, we're actually going to pipette the transfection mix prior to adding the cell suspension, whereas in our forward transfection, you would transfer the cell suspension and then the transfection mix. So here, he's pipetting the negative control, which just has the Optimus buffer, or the Optimus reagent and the jet buffer. And now he's going to add 50 microliters of the positive control declover using a new pipette tip into the declover well. And it's always important to change pipette tips as we don't want any contamination or mix crossing of our transfection mixes. And it's always important to make sure you pipette the mix evenly within the well so you get a better distribution across all cells. And now he's just grabbing the Pnex MTS and pipetting it into the fourth and final well for the transfection. Here he showed us the cell dilution that we made right at the beginning of the experiment, and he is using a P1000 pipette and is pipetting 500 microliters into each of the respective wells. And it's important to change tips here as well, as we don't want to worry about getting DNA in certain wells and cross-contaminating them. And it's important to make sure the pipette tip is also in the cell suspension, as we don't want to get air bubbles and we don't want to lose the full 500 microliter amount that we need. Once we pipetted the mix and the cell suspension, we can now put it in the incubator at 37 degrees Celsius overnight, and we can actually look at them under the Zoe imager the next day or whenever you're back in lab. There you go. So now that we've run the reverse transfection protocol and looked at our each of our wells and or slides, depending if you mounted the cells underneath the Zoe and got images to see if our transfection worked and to determine the percent transfection. We're just going to show you some examples of the transfection, specifically the one we did, and your expected results. So here we have the negative controlled merged image with no transfection DNA. So ideally we should see nothing. So this very weird hazy green, this is actually the bright field stain, the bright field of the HeLa cells. So there's no DNA. We can see that here, but we do see, obviously we do see the blue dots and the little green speckles. So that's from the Hostin 48 phalloidin dye stains, which were added after the transfection. And these are just other ways to look at particular organelles or look at the DNA within the cells. So this is just a merged image of them. So the blue dots is a host stain, and then the green dots plus the background is actually phalloidin. Here is our positive control, also the stained HeLa CCl2 cells with declover. And then we also have host staining here, which is the blue dots. So the blue dots host staining just stains DNA. And this is actually one thing you could use to actually count the total number of cells within your sample. However, there can be issues involving whether or not what phase these cells are in mitosis. So maybe we see the sister chromatin that are coming apart during mitosis, and sometimes you can count maybe one cell as two because they're undergoing that step. But it's a good way to look at the total cell count, or you could also use a bright field image as well, like in the negative control that we showed. And then the green stain on the left is the declover stain, which actually just stains the cytosol. And then on the right, we have a merged image of the two. And these were analyzed using ImageJ, which you'll get more familiar with throughout the semester. So for our total cell count using ImageJ, for the positive control declover, we actually got a total count of 116 cells using uh, using the threshold, adjusting the threshold and adjusting the contrast of the image using the built-in cell counter in ImageJ. And then counting the total number of cells as well, we actually got a percent transfection of 25.6%. And this was done using ImageJ. So here's an example of our more experimental transfection, and this was 3x MTS or NLS. It's the PLEX plasmid DNA. So here we can actually see images of the mitochondria. So that's what the DNA targets. Here's an up close image in C, and this is just HeLa CCL2 cells stained with the 3x MTS I, which is also the PLEX. And here you can see the mitochondria. These are very clear images taken on the Zoe. And then A and B are split images of a merged image. A is, again, the host DNA stain, and B is the HeLa CCL2 cells stained with the scarlet from the experiment. And the total number of cell counts we actually got from this was 23, and that gave us actually a percent transfection of 3.9, which is generally really low. So there could be a variety of issues that come up within your experiment that could result in that, or even it could just be the plane of field you're in when you take the picture in using the Zoe imager. And that is our general results.